So thank you. I think I'm ready to go. So I'm Kuba. I'll be telling about the project that we've been working since I think 3 years. So as Mike said, we wanted to have a small team within a big company, very experienced people, very motivated, and with some university background. So we all know machine learning and we all know image processing. So that was mostly medical imaging, so that's where we started. And we end up having an idea for an application, not for a product yet, but for an application, so it was dynamic imaging. So I will talk about ECONIB, so that actually means something. If we have a look at the grant name, grant application, we'll see that we are interested in dynamic contrast enhanced imaging. So what we want to do is to extract some value from that kind of imaging. So basically we are interested in the fourth dimension, in time, because we are injecting contrast. This contrast goes through the tissue and we want to see the profile of this contrast in a specific part of the body. So here we were actually focusing on gliomas, so it's brain imaging, so MRI of brain. So if we look at dynamic imaging, we will see that it's already being analyzed, so there are some steps to be performed and we know actually all the steps. So why not to automate the steps and make the life easier for clinicians or for the medical physicists that actually do that kind of processing. So we started from the original scan, MRI scan, and we want to produce the final report which contains the numbers. So we want to somehow quantify the contrast flow within the tissue of brain, for instance, or any other organ. So that was the idea, because we want to develop the technology to be able to solve that kind of problem. And we looked for a clinical partner. So that's our clinical partner from our place, from Poland. They are quite well known in Poland, very well established center of oncology. So they are providing us with data and like interpretation of the numbers, what they mean and what they actually would like to be interested in achieving after finishing this project. So here we can have a look at a workflow. So after acquiring a scan for a given patient, we need to perform a number of steps to produce the final value from that kind of imaging. So first we need to somehow store the image data, so that's usually done in PACS, in a hospital image system. So it's a sort of database. Then we can somehow download the data and try to process the data to extract the numbers. And here where we are. So after acquiring the data, we want to process the data and we want to do it in place, so in the hospital. No transfer of data, as Lawrence asked Alex. We are not interested in transferring data because it's basically difficult in that kind of scenario. And finally, after this automated analysis, we want to have a report that might be read anywhere, anytime. So we have a DICOM report that will store the results. But it's not only the final results, so it's not only one single number, but the results that we achieved during the processing pipeline. So that might be, for instance, segmentation. And finally, if we have this report, we may store it back in PACS, or we may analyze it locally and just not push back to PACS. So we are interested in contrast dynamic enhanced imaging, the profile of that kind of analysis. So we start from the MRI study for brain in that case, and we need to perform a couple of steps, as I mentioned. So first we need to segment the brain tumors from brain imaging. That's the part of the brain that we are interested in. And additionally, we need to extract this vascular input region that pumps the contrast into the tissue. After that, we may have modeling of contrast flow in the tissue, and after that we can quantify the curves of the contrast using some numbers that are already established. They are used in the literature, so we know that they actually work. And we can finally build a report that contains all the valuable information that might be used and that hopefully will be used in clinical practice. So let's go through a quite simple example. So we are given a scan and we are interested in that region. So we know that there is something that might be glioma or any other tumor in brain. So we want to extract this region automatically. So as a backend here, we have deep learning. So that process is actually a two-step process. First, we need to detect the tumor within the scan of the brain, and second, we need to segment, so find the boundaries of the tumor within a given scan. So here are units. So this U-type architecture of deep networks, and we have a training set which is composed from data which is acquired in our site. So our clinical partner delivered us data and grant through segmentation. So we know what should be the real segmentation, and we can build a model based on that data. It's data-driven, as we discussed today, so it has advantages and disadvantages, but that's the way we went in this project. I think we can somehow prove when and how it works. So basically that was the first step, to find the tumor within the brain. The second step is to find this small vascular area that will pump the contrast into the tissue. And given those two, here's the 3D visualization of this part. This guy. So having those two, we may have the profiles of the contrast flow in time. So on the x-axis, we'll have time for both vascular input region and the tissue. So the tissue is tumor in the brain. And we have contrast concentration on the y-axis. So given those points, we may somehow approximate the curve to fit those points in time. And those curves can be actually described by two numbers, so we can have, quantify the curves with numbers, and those numbers are easier to interpret and compare against each other if we have only two numbers on the two curves. So this curve is for this vascular input region, and the second is for tissue. So you'll see that the curve is different, the values are different, and we can have a look at the shape of the curve, which actually is meaningful. So that's the shape which changes, and that's important to know how, for instance, the patient responds to the treatment. So we need to be able to monitor and somehow quantify and compare the curves extracted for a given patient in time. So that should be robust. And here's the example of how we can visualize the numbers. So if we extract just two numbers, which is K-trans and V, we can have the visualization for every single voxel in the image. So that might be not the best example, but the tumor is somewhere over here. So those values should be somehow important. So they should somehow quantify the characteristics of the tumor. It differs for different tumors, for different stages. However, that's the way it works. So if we have dynamic imaging, we are interested in those two numbers, and we want to analyze those parts of the image. And we may even compare with different parts of the brain which are healthy. And here's the final report. So as I said, we may have the segmentation and parametric map, so K-trans, VE, original data, and the curves. And the curves will also be quantified, so we'll have a table of numbers saying what is the characteristic of the curve. So that is what we can push back to PACS or store locally to analyze further. So if we think about this application, we may think of a number of advantages. First, it's fully automated. So we have a scan of patient, we can push it to our system, and we can have the results. So we will have a report summarizing all the findings and all the value that you can extract from that kind of imaging. So we don't have any user interaction. So we just push data and get the result. And of course, if we can automate this process, we'll make it faster. So as compared with manual segmentation, manual analysis of that kind of data, I think it takes around 40 minutes in our local hospital compared to less than 3 minutes using this application. So it's not a product, it's the application. So we actually want to make life easier for clinicians. We don't want them to not find something in brain that is suspicious, so we want to detect everything that should be detected. And we want to actually seamlessly integrate with software that's already in there, so that should be easy. And that was the part of what we are trying to achieve, and the second part is how we are trying to achieve that. So I would say it's very different from pure research, from basic research, and from development of any other application which is not medical device. So I want to focus on basically two things. First is the research part, because that was R&D project, so we are interested also in the research part. And I think that's like the pivotal part of any project that involves research. So we need to be able to reproduce the numbers anytime, anywhere, with the same algorithm and with the same data. So I think everyone knows how it works in practice. So first we are posing a question, so we have a hypothesis that we want to test. So that's the first part. We design the experiments and we run the experiments over a given data and over a given implementation of algorithms, and we collect some artifacts. Once the artifacts are collected, we may analyze them and try to like infer some knowledge from the artifacts from the experiments. So yes, first we have a hypothesis, we want to experiment with the hypothesis, we want to check whether it's true or not. Then we run the experiments and we store the values from the experiments in one place. And finally we want to analyze our observations and see whether we need to tune the algorithm or we need to maybe have a bigger sample of data to make it provable. And we are in a company, so we need to actually be aware of the research process within the company, which might be a bit different from the research process at the university, for instance. So here we know that new numbers may come from very different sources, so we may have new algorithms, new implementation, we may have corrected some bugs, or we may just change some minor tweaks in the algorithm, and that will produce different numbers. So there are a bunch of different sources of new data that we need to analyze. And we need to remember that it's fairly difficult to analyze it properly, because we may very easily generate a big amount of data that will be multimodal, very difficult to analyze and compare. And also, if we have some people that are not very experienced in research in the team, that might cause problems, because they will store different things, maybe differently, so it will not be trivial to compare with each other, and that might be easily a nightmare, and definitely is something that we want to avoid. So what we wanted to design is a hands-free research workflow. So basically we want to be able to design the experiment, implement the experiment, run and store the values without any interaction. Sounds good, but practice is different. So we wanted to make sure that we take advantage of tools that are already in there. So we have a company, we have some good tools that are already in use, and they are actually used worldwide, so everyone uses that kind of tooling. And we actually found some parts that we need to have in order to have that kind of research workflow. So first we need to be able to run experiments seamlessly, because we may have different machines with different architecture, different hardware, for instance GPUs. Second, we need to somehow store the code, which is a bit obvious, so we need to have a versioning system to store the code and to be able to track back to a given version of the code. And finally, we need to have a place where we should store the artifacts. So basically, here's the place where we store the results. We can dive into the numbers and image data and see what we have from the experiments. And we can observe that those three things are fairly basic. So everyone in industry uses continuous integration servers. Everyone uses source control, so it might be Git, SVN, whatever. And everyone uses some space to store. So it might be Windows shared drive or whatever, but there should be a place that is accessible and we can like easily access and read data from this place. So we ended up having that kind of processing pipeline. So we design the experiments, we launch the experiments, and we create a continuous integration job in the server. So we know that the server knows that that experiment should be run. We know a list of hardware where we can run the experiment, and we have a queuing system just to spawn the experiment into a given platform. So after that, it's all done automatically. So we run the experiment in a given machine with a given hardware. For instance, for segmentation, we use GPUs. And then we collect and publish artifacts. So we store, for instance, images, numbers, Excels, whatever. And we can send the feedback to the guy who was running the experiment. So this experiment is finished. You can have a look at the numbers and maybe tweak or change the experiment, run it again, and see whether it helps or not. So I think the cool thing about this pipeline is that it's totally automatic. So you just say that you want to run this experiment, and it's done like in an automatic way, using only the tooling that is always available in any software company. So it's not black magic here. So yes, as I mentioned, if we have continuous integration, we may easily have a queuing system where we queue the experiments to run. Then we can select the hardware where to run those experiments. And I think what is also important is that after finishing the experiment, we'll have the result like in an automatic way. So we can get an email saying that this experiment finished. It may take like, for instance, 1 day, so it's not super fast. But after that, we may have a look at the results. So we don't have to disrupt ourselves from other work. We may just wait till the results are in there and ready to be analyzed. And of course, source control allows us to store the versioning of the code, so we can easily trace back to a given version and see what happened between the current and the previous version to see what was the impact of the changes. And finally, in the remote storage, we may have all artifacts in one place, or are like stored in the exactly same way, so we are avoiding problems with interpreting the artifacts. All is exactly in the same place, the same way of storing, so that's also important. And also, Alex already mentioned that, so regulatory is very difficult, it's time-consuming and very costly. I want to focus only on one part, which is risk management. Because in academia, if we think about risks, we may think about the risks that are coupled with algorithms. So you may have a risk of not delivering state-of-the-art numbers, but it's not only the case in that kind of setting, where we're trying to develop a product, not the algorithm. So basically, we may divide all the risks that appear in that kind of project into 3 subgroups, or even more, if you like. So first, we'll have technical risks. So that's exactly what I mentioned. So actually, it's not only algorithms, but also, for instance, implementation. So we need to be aware that the implementation should be robust, should be scalable. So if we have more data, it should be executed in a seamless way. And of course, if we take something from the literature, it should report the numbers that are being reported in the literature. So it's not always the case, and we need to be able to find why it's happening. So those are risks that are connected with algorithms and the technology itself, but it's only a tip of the iceberg. So we have also management and business risks. So in the case of management risks, we have a fairly experienced team, but it may change in time. So if someone leaves the company, we need to find a person that might replace this guy in a short time, which is very challenging at the moment. I think not only in Poland, but probably worldwide. And finally, we were trying to build an application, and from the application to the product is a long journey. As Alex mentioned, it was a very good example. So we may have a very good application that works like in a perfect way. So we have state-of-the-art numbers, but if it's not usable for clinicians, like there's no value in using this thing. So I think this value might be on different levels. So if we can optimize the time spent on analyzing the data, so if we can optimize the time of clinician, then it's also a big value in clinical practice. And also if we can couple the numbers that are already in there with some new numbers, like maybe texture or any other quantification of data, then it might be another way of giving the value to clinicians. And here's the example on how to assess that kind of risk. So we may have the probability of appearing the risk, so we can see that some risks may be rare and some will almost always occur. And we have the consequence. So if the consequence is high, we can't afford that kind of risks, because ultimately it may affect the patient, which is definitely not what we are trying to do. So basically we should operate in that part of the table. So if we are low and medium, then it should be fine. But we need to remember about monitoring that kind of things in the project that involves medical software. And here's the example on how to quantify the risks. So here is an example of management risk. We'll see that in practice we have two parts of the table. So first we'll have the action which should be undertaken in order to mitigate the risk. And we will have three things: the likelihood, so the probability of this risk to happen. We'll have the impact on the project, and we'll have the risk level, which maps those two into one number or one letter. So in practice, what we usually do is to either decrease likelihood or decrease the impact, or in the ideal world, both. So if we are decreasing this level of risk from medium to low, then it's fine. But what's important, we need to monitor those risks during the entire project. So from the very beginning till the very end, we should keep in mind that the risks are everywhere, and we should not forget about them. So I think it was a big step to the company, because we were cooperating with Mike on quite small projects. Now we were working on a big project for 3 years, so we built some experience in team. So basically we had only 2 people. Now we have more than 2 which are experienced, and I think they are able to attack and solve any problem at hand. And what is also, I think, important is to remember that we are trying to develop the application, but if we are trying to build a good architecture, we may divide the entire application into modules. So we may have a good segmentation, which might be easy to change in the future. We may have good modeling of contrast flow, which also might be changed in the future, and I think that should be the way to go, to have a good pipeline with some parts that might be easily changed if necessary. So now we are going to RSNA this year, so we'll face with clinicians in that kind of real setting. I think we are in a process of making a product, so we have a good application and we want to make it a real product that will be used in clinical practice, hopefully, but we need to see this value in practice. So that's it. Thank you for the attention. I'm happy to take questions.
Thank you, Jakub. Any immediate questions? Or facts?
It's a bit boring. What advice would you give to people making medical imaging software from a development point of view? Should they do test-driven design? How should they use GitLab? And what would you say to someone that wasn't doing any of that?
I think it depends. It very depends on the problem, because I think test-driven is always good, but you need to be sure that it fits your application. So for instance, if you develop like segmentation algorithm, it might be difficult to apply. So definitely I would advise to use that kind of toolkit. So we need to have Git, we need to have like very well established pipeline for experimentation, because otherwise we are not able to trace back to a given experiment and check whether it worked or not and why. I think that's the more important question. We need to be able to know why it worked or why it did not work. So that would be my advice.
A question both to you and probably to Alex. How do you control for the input data in the first place? Are you advising the hospitals on the optimal data acquisition method?
Very good question. So for now we use only Siemens data, so it's very narrowed.
There are other factors available.
Yeah, no, I think the question. No, I think it would be. I think it would be difficult in practice, because if we are not sure that data will fit the system, we should not produce any numbers to make sure that we are not generating numbers that cannot be interpreted. So that would be the first step of the processing pipeline, to see whether data fits our assumptions and if the data is correct, although it might be correct in a different way. So you may have different scanner which produces different data which is still correct, but it's not correct in terms of our software. So we need to be able to make sure that we only analyze that bit of information that we can analyze. Otherwise it will...
You think you can do that? Then you can
it's very difficult.
QC as the data comes in and reject image quality reasons or acquisition, if it's the temporal resolution is too poor.
I think that might be the second step, as also Alex said during the last presentation, is that multi-site analysis. So if we have different hospitals around the world, we may try to collect the data or give them the application and see what the values are. So I think it would be extremely difficult to prove that we can work with any data, but that will probably take time.
As a small company, that's sorry.
You're my official party line.
Yeah, then the unofficial, what you really think.
No, so actually, I've got quite a good answer for this. So first of all, we work with image registration. So one of the required DICOM header bits of information is voxel size, and we can measure the size of that voxel, or sorry, the movement of that voxel in mils. So we use an SI unit, and that actually quite nicely standardizes us across different platforms. So you find there's actually quite low variation where we'd expect to see low variation, because we're basing on size. I think it's more difficult when you use signal intensity, for example, ATC, diffusion, enhancement, perfusion, all that stuff is very bound to the intrinsic nature of the coil and the magnet. So I think we've got a fairly good answer to that one. The second part of it is our intended function is really about tracking over time. The person is probably their own control, and you basically look at the time point 1 versus the time point 2 scan, and that also helps to kind of force a degree of kind of comparison to kind of historical. I think we are going to run into trouble when it comes to just pulling out a random number at a point in time and expecting to see that work as a kind of standing biomarker. And just as one final point, so prostate-specific antigen for prostate cancer, doesn't work very well as a point test. I think it's used like that in clinic. But they have found a lot of utility in tracking PSA over time, and I could see a lot of imaging biomarkers being used in this longitudinal fashion, how they trend, and I think that's just kind of how clinicians use it. And I think this is what clinicians might want to do with it and help that guide the validation process.
It's for a small company breaking into quantum imaging space. It's difficult. We're not Siemens, we're not GE, so we can't say only people with our scanner can do this or whatever. We have the same experience with TextLab. You have to start somewhere. You have to build your first product. You have to get into a hospital, work with them, and then expand. And I'm part of why I wanted to bring these presentations to the industry session is the route. If one of you out there has an idea, you have something you think want to get out there. It's quite complex. It's exciting. It's interesting, and occasionally boring. But it does have some challenges you won't necessarily have met in the academic world. It's worth talking to people, talk to Kuba, talk to me if you're here today about the experience. We'll try not to put you off. But it's a complex journey. You end up learning an awful lot as part of that. And you have to develop relationships with clinicians. Some projects start with a single clinician firing off and then you have a partner. But sometimes you have to actually get the work done and get people to adopt it. There's a variety. Another, yes?
Yeah. So I was going to say, Kuba as well, but effect size. If you've got a tumour that weighs 3 kilos or something like that, and it's changed by a millimetre cube. No one cares. Like the actual accuracy of the algorithm in those extreme cases isn't that big a deal. How accurate you are. And I kind of wondered, for us, our effect size is probably about 0.1 unit either way. You know, that's where we need to be good at. But someone redoing our algorithms and getting a marginal increase on that doesn't affect the actual utility of how that thing will be deployed in clinic. How have you dealt with the effect size question? I know you've entered a competition at MICI recently, and you know they're all grouping up quite close together.
Yeah, no, it's a good question, especially in this brain tumor segmentation, because we may have different algorithms which give very small differences. So they are good, they are robust, but they are slightly different. So the question would be how the segmentation will affect the curves, the constant concentration curves. We didn't check that. I presume it will not have a big impact because we look at 3D, so that might be of minimal impact, but we didn't check that. So.
I mean, ultimately, the effect is the biological end effect, right? I mean, because I mean, you are trying to answer the question as if it's an imaging problem. And you can debate for ages about whether small changes of the shape is relevant or not. But at the end of the day, it's the treatment outcome, right? So it's a decision-making process.
Yes.
And if the changes doesn't change the decision-making, it doesn't matter.
Yes, exactly.
So it has to be coupled to a medical underlying question. Which type of cancer is it? Is it palliative? Is it curative? God knows what. I mean, so it's not, you leave the domain of image processing the moment you talk about, let's say, the effect or yeah.
And a fundamental question as a company is, will we make money out of it? But the next step down, going to the medical environment, is will it be useful to a doctor? If it's not useful to a doctor, we're not going to make money out of it. And so it has to actually change the treatment of the patient, or be very useful in research, which most of these projects will start off with a research grants to find out something. And pure knowledge, motility turns out to be useful, but even if it wasn't, understanding the motility of the gut is interesting, and there will be people who are interested in doing that. Any further questions? Otherwise, I think we might be having an early tea break. Okay, well, thank you very much. If we could thank our speakers.